Hello friends, welcome to BMH Learning. In this video, I'm going to talk about ATAC sequencing, also called ATAC-seq. So, ATAC stands for Assay for Transposase Accessible Chromatin Using Sequencing. It's a technique used in molecular biology to assess genome-wide chromatin accessibility. So, by sequencing regions of open chromatin, ATAC-seq can help you uncover how chromatin packaging and other factors affect gene expression. Now, there is something important about this method which make it different and more powerful than its counterparts. The technique does not require prior knowledge of regulatory elements, and this specialty makes it a powerful epigenetic discovery tool. It's much better alternative when it comes to effective understanding of chromatin accessibility, transcription factor binding, and also gene regulation in complex diseases, embryonic development, T-cell activation, and cancer. Apart from these, ATAC-seq can be performed on both bulk cell populations and as well as on single cells at high resolution. Let's get into the concept of chromatin accessibility. What is a nucleosome? Have a look at the diagram. This is our DNA. Basically, the DNA in the nucleus is not present in this way. Instead, it's wrapped around histone protein complexes. So when the DNA wraps around histone, we get nucleosome. Now, as you can figure out, the DNA present in a nucleosome is tightly packed. So this type of DNA is considered mostly inactive. So we can say a nucleosome actually limits the accessibility of the DNA. On the other hand, if the histone proteins are removed, the DNA becomes free and thus active. So the unwrapped DNA is more accessible for transcription. So the genes in the open region or histone relieved region are transcriptionally more active. Now let's know, how does ATAC-seq work? First step is the cell preparation. First we need to harvest the cell. Now, counting of the cells is very important. It's because the number of cells used in ATAC-seq assays is very crucial for the transposition reaction and size distribution of the generated DNA fragments. Moreover, cells should be intact and in a homogeneous single cell suspension. Cells are lysed with a non-ionic detergent to yield pure nuclei. Next is transposition reaction and tegmentation. So in this step, genomic DNA is exposed to TN5, a highly active transposase. The TN5 binds to the accessible region of DNA. So, as we know, TN5 is a transposase. Its work can be defined as cut and paste mechanism. The transposase fragments only the accessible region and sequencing adapters are added into these regions. In the next step, we are going to amplify the tagged DNA fragments. First we follow the extension step, which generates double-stranded template with the tags at both ends. And for the amplification, special primers are used which contain a barcode sequence. So, all the fragments in the genome which were accessible by the transposase are now amplified. Now, these amplified fragments contain sequences which are required for next-gen sequencing. Next-gen sequencing will reveal which regions in the genome were actually accessible. This was all. Thanks for watching.